All right, in the next few videos, we're going to be talking about drugs, right? So drugs that we develop to treat infectious disease. So the origins of most antimicrobial drugs are coming from other organisms. So we talked about the discovery of penicillin and Alexander Fleming, and you talked about exotoxins and exoenzymes that are produced by certain bacteria to compete in their environment. So now it should be pretty much known or easy to understand how and why, bacteria produce these antimicrobial compounds, or molds, or even plants are producing antimicrobial compounds. Some common ones that you may be familiar with, just even the names of them will probably remind you of some drugs that you've heard about, some antibiotics that you've heard about, and ones that we will use in the lab. So bacteria, Streptomyces and Bacillus, molds, Penicillium and Cephalosporum. So these are organisms that are producing antimicrobial compounds. Now, once we identify the antimicrobial compound that's being made, naturally in the environment by these organisms, it doesn't just then directly translate into a drug. Most of our drugs are said to be semi-synthetic, which means they're naturally occurring, but then we take them into a lab and we manipulate them, add different groups here, and we make them into a good functional drug. But it starts with that baseline of finding the natural occurring antimicrobial compound and then sort of semi-synthesizing it so it'll work. So the goal of any antimicrobial drug is to, inhibit the growth or to kill either bacterial eukaryotic pathogens or block viral replication. Typically we're going to be inhibiting enzymes that are involved in all of those functions, the formation of large macromolecules, peptidoglycan, proteins, and things like that. And I'll go through those in a second, sort of the major modes of action of these antimicrobial drugs. The other thing that we want to make sure is that the drug is selectively toxic. And what that means is that it's not going to do damage to the host, right? So it's not going to, if we're going to take that antibiotic and try and treat an infectious disease that we're infected with, right, we want to make sure that it does little to no harm to us, but is only, selectively toxic to the microbe. Now, depending on the kind of drug, we can have drugs that are considered either broad spectrum or narrow spectrum. So narrow spectrum means that those drugs are really only inhibiting or killing a very small group of bacteria or organisms. Broad spectrum is they affect more, right? Now, ideally, we want them to be as narrow spectrum as possible so that we're really only attacking whatever's making you sick. But we don't always know the source of an infection. We've talked about that through the discussion of immunology, and infectious disease, that sometimes it can be a little tricky to know exactly what someone is sick from, and it might be easier to use a broad spectrum antibiotic that is works on inhibiting the growth or killing more types of organisms. So let's get into the different modes of action of how do drugs work. So the most common are the first three, where we're going to have ones that inhibit cell wall synthesis. So the formation of that peptidoglycan layer. They are going to inhibit the function of nucleic acids, right? Or the structure of the building of nucleic acids, or they are disrupting protein function or the formation of proteins, right? Now, some other ones that are less common, and we'll talk why they're sort of special types of drugs, are ones that attack a pathway to make folic acid, and then also interfering with cell membrane structure. All right, so these are the major modes of action of different antimicrobial antibiotics. So there are three major classes of bactericidal antibiotics. The first one is called, is a group called beta-lactams, and these are ones that are attacking that cell wall. So that's their mode of action, is disrupting the peptidoglycan. And some examples of that are penicillins and cephalosporins, that are going to inhibit enzymes that are responsible for the formation of that peptidoglycan layer, which is a very important structural component. We know why, peptidoglycan is so significant. The second major class of antibiotics is called fluoroquinolones, also known as quinolones. And these are ones that are disrupting the nucleic acids, right? So they might be blocking nucleotide formation, inhibiting replication, transcription, inhibiting the formation of DNA synthesis, right? Some examples are norfloxacin or ciprofloxacin. And I think Cipro is one that we'll be using or looking at the results in the lab. So we'll be looking at one of these kinds. Right? And then the third major group is called an aminoglycoside. And aminoglycosides are the one that attack the proteins or block protein synthesis. So they can inhibit translation. They can interact with the ribosome mRNA complex. So when we went through genetics, we sort of went through how that messenger RNA docks on the ribosome in order to form, this primary sequence of amino acids that goes into forming a protein, right? So this is the mode of action of aminoglycosides. And some of these drugs probably sound familiar to you, tetracycline, chloramphenicol. I'm pretty sure we use both of those in the lab, and also erythromycin. Okay, so those are the three most common ones, but then we have other ones that aren't as common. So disrupting cell membrane function. So remember that the cell membrane of a bacteria is very similar to the cell membrane of a eukaryotic cell, which is why it's not really a great drug target. But there are drugs that we do have that select for very different or small differences in the bacterial membrane from the eukaryotic membranes and different lipids and things that are in the cell membranes. So there is the ability to create drugs that will damage the membranes of bacterial cells. Now, folic acid synthesis. This is a very special sort of pathway because eukaryotes do not synthesize folic acid. This is specific only to bacteria, right? Only microbes can perform folic acid synthesis. Folic acid is an essential compound that is needed to synthesize all sorts of major components within the cell. Purines and pyrimidines that make up nucleic acids, and amino acids that make up proteins. So it's super important compound. However, folic acid, it needs to be consumed by people. So we get it by consuming it in our food, but it's actually synthesized by microbes. So the fact that there's this very special pathway to make this, that's something different from what eukaryotes do, it creates a really good target for us to develop a drug for a pathway that's specific only to bacteria. So this is a rare case in which we have, studied this pathway in depth and have developed a drug that is actually competitively inhibits the active site of these different enzymes that are involved in this pathway. So most other drugs are going to bind somewhere on an enzyme that are going to sort of change its shape. They're not going to be what is called a metabolic analog, where it's a shape that is going to bind straight into the active site and prevent the formation or the active site from binding the substrate. So some examples of that are sulfonamides, trimethoprim, right, are ones that block folic acid synthesis. And again, here, just so you have an idea of what some of these things are used for, right? So sulfonamides are sulfa drugs. You probably may have heard that. They're considered the very first modern antibiotic. They're completely synthetic, like I said, so they're designed to be a metabolic analog to bind to that substrate, because we were able to study that very specific pathway that we know that humans or eukaryotes don't have. Used for UTIs, trimethoprim is used to treat pneumonia in AIDS patients. Sulfones, another kind of folic acid synthesis targeting drug, is used to treat leprosy. So just an idea of some of those, what those drugs are for. So up till now, we've been talking about drugs that are antibiotics. Antibiotics are drugs that kill bacteria, right? So they are not useful on any eukaryotic pathogen or on viruses. We do have some drugs that are antifungal drugs that are going to attack eukaryotic pathogens. However, one of the major problems that we have with this kind of drug development is that it's difficult because of how closely related these organisms are to us, right? It's difficult to design drugs that are selectively toxic only to fungi. So we tend to have more side effects that arise because of the similarity between the eukaryotic pathogen and the eukaryotic host. But we are able to identify some fungal proteins that are different than ours that we can, design drugs that will work on them. But again, tend to have a little bit more side effects, a little bit more difficult drug development because of the close relation. It gets even worse when we're talking about helminths, right? Worm infections. So they have they're way more advanced organisms, so they're multicellular and they're even more similar to us. So most of the drugs that we design to work against helminthic infections are ones that are going to block the reproduction of the adult worms or try to immobilize or break down the worms or get the worms to be expelled from the body, right? But still pretty difficult drug development because of how close they are. Now, on the other extreme of things, we have viruses, right? So viruses, as we know, are very simple and there's almost nothing to them, right? So on the other end of the spectrum, they're so far away from humans, right? There's not a ton of similarities, but they don't really do much. They hijack human host machinery in order to do pretty much everything that they need to do within the cell. So it's difficult to develop drugs because they would be impacting our own cellular processes. So drugs that are antiviral drugs work to help to prevent viruses from getting into the host cell. There are some viruses we know that have specific enzymes, specific only to them that they carry around. But that's only certain groups of a virus, or certain families of virus. So for those, we might be able to develop drugs that would inhibit reverse transcriptase, if you remember from our virus lecture, or somehow preventing the maturation of the viral particles. But again, very challenging, and really the best way to prevent a viral infection is through vaccination. All right, another thing that we need to think about when dealing with drugs is no matter what, if you're putting a drug compound into your body, it is potentially toxic. So how much drug can you take? And over how long of a period of time? There is always a potential hazard for doing damage to tissues and, major organs. Also, there can be allergic responses. So if a person responds to a drug like it's an antigen, it can stimulate an immune response. I think I'm going to stop there, and I will finish up the end of the lecture in the next short video. We'll talk about how drugs can affect your biota, and then how to determine the type of drugs we use for what and tie it into the lab that we're going to be doing on antibiotics.